should have reinforced what we did with the HHMI activity, should have reinforced what I did with the lecture. Just a different. The one thing I'm seeing with the MyAP videos is they tend to skim a little bit. They tend to not get into some of the nitty gritty. Anything? Once, twice? Okay. So we are going to go to, in a moment, to pivot, but I want to show you, I want to walk you through a few pieces so you're not hopefully confused as you try to work through it. It's actually a pretty short pivot. And so what I'm going to do is I'm going to put you into your, once we get going, I'm going to put you into your teams in a breakout room, and you can work together in your breakout room to complete this. Something I think you can complete in class in about 30 minutes, I believe, is my guess. Okay. So first thing I'm going to do is in the chat, I'm going to put a link to the assignment. So give me just a moment. Coming back over. Okay. So if you check your chat, there should be a link that should take you to an activity called Introduction to Photosynthesis. So go ahead and open that up. But don't start it. I want to walk through a couple things with you to hopefully avoid any problems. So this first activity looks at the basics of the two primary gases that are involved of carbon dioxide and oxygen. When are we taking carbon dioxide in? When are we taking, where's the oxygen going in or out? Is the carbon dioxide going in and out? How does the plant respond when there's light versus dark? Is sort of the situation we're looking at. Did the link work? Yes? Okay, so let me show you from my end. Okay. I'm going to leave this page and just go back so I can show it to you. I'm just going to go into the preview mode. So the basic directions are right here. It's a little bit of a pain. Took me a moment, at least took me a moment to figure out exactly what they wanted me to do. So they're using what's called a Vernier probe. Vernier is a company, and we're using two different probes. We're using a carbon dioxide probe, and we're using an oxygen probe. These two probes are sensitive probes that are connected to this phone. One of the probes is measuring the CO2 in parts per million, and the other is measuring the gas in percentage, oxygen in percentage. So those are the two things that we're looking at. You're going to open up the tools up here, and you're going to open the timer. And this is just to reset the timer if you need to. And then it says so try to stay with me because this is the part you need to know. You're going to start it just for the setup. So they're taking a regular basic plant. They're sticking it into this closed chamber. And once they get it into the chamber, you're going to stop. You're going to stop it. You can see here's the two probes. I don't know how well you can see them on your screen, but those are the two probes. And what you want to do at this point is reset the clock to 0. So just hit that and reset the clock to 0. All right. So once the lid is put on, hit the pause button, reset the clock to 0. Here's my initial reading. So that'll be your reading at 0. So down here in the chart, I'm going to put the CO2 levels are 738. And I'm going to put the O2 levels were 19.61. It's going to convert that to parts per million automatically in the last column. All right. So this is the percent. That's the parts per million. And this is happening in the light. This is happening in the light. Okay. certain times in the video, it's going to go dark. So you want to record, was this taken during the time the lights are on, or was this taken during the time the lights are off? So obviously if the lights are on, you're going to put light. If the lights are off, you're going to put dark. Capiche? Okay. So what you want to do to get the readings, and this moves pretty quick. Okay. It's in minutes, but it's time lapsed. It's time lapsed. I want to stop at about every 10 minutes. So what I'm going to do is I'm going to hit the play button, and I'm going to be ready really quick to pause it. If I say 3, 4, 5, 6, 7, 8, 9, boom, not too bad. 10 minutes. I can, if I overshoot it, I can just take the slider back and I can get it to be around the same right time. I don't have to get it exactly at 10.0, but get it pretty close to 10 minutes. Yes or no? Then I'm going to take and record those numbers, 692 and 19.64. Whoops, 692 and 19.64, and that's in the light. Oops, I accidentally put a comma there. All right. And so that's how I'm going to continue until I've done about 120 minutes, I'm sorry, of data. So again, what I would do is just pick up, and I'm watching it so I can stop it somewhere around 20. Not too bad. I would record those numbers and continue. At some point, I'll just slide it forward. At some point, it will turn dark. So here it's pretty obvious the lights are off, so you're going to record that it's dark. Am I making sense still? Yay, nay? All right, so you're each going to fill your own out, but you can work together. You can collect the data together, yada, yada, yada. That data you're going to graph down here. Right. Remember, you can change what you put in here. You can add additional factors by adding columns that'll put more things on the y-axis. You'll create the one graph, and then there's about 7, 8 questions, and then this part 2 is basically 2 questions. Right, so this one's fairly short, and it takes us a look at the basics of photosynthesis. Okay, so I'm going to stop sharing that for a moment. Questions? Okay, so it'll take me a second to organize you into your groups. Give me just a moment here. What happened to you? Just a second, I gotta bring it back up. Oh crap, I can't get to it. Oh wait, looks like Google might be working. Let me try. I believe Google is back up. All right, cool. Anyway, I'm going to be putting you into your groups. Give me just a second. Google Meet, breakout. Sorry, one moment. There we go. And not quite working. All right, so this might take me a second longer. Partially working. False positive there. Okay, so hopefully you know your group. I'm just going to have whoever the captain or spokesperson is speak up in a second. If your quote captain spokesperson is not present, then somebody else speak up. Okay. So who's number 1?
Me, Holden, Peter, and Carter.
So Bryce, Holden, Peter, and let's see, Carter's here somewhere. There he is. Okay. Room number 2.
Me, David, and Mike.
Was that Alan speaking?
Me, yeah.
Okay. And David
David and Michael.
Rancilio, I'm assuming.
Yes.
All right. And there's Alan. Beautiful. And then number 3. Who's number 3?
It's me, Hannah, Paige, and Mara.
Hannah's here. Paige just texted me and said she can't get through on Zoom. All right. So I'm recording this. I will put a recording out for those that couldn't make it. All right. So there's 3 of you here. And then room 4. Who's group 4?
It's me, Mac, Jack, and Frank. But it's just me and Mac in the meet, I think.
All right. Who was speaking? I couldn't see.
Ethan.
Ethan. Thank you, Ethan. Okay. So it's just you and Mac, you think?
Yeah.
Okay. And then room number 5.
It's me, Cass, Aiden, and Jess.
Aiden and Jess. You actually have everybody.
And Cass too, yeah.
Okay. And then room 6.
That's me, Aaron, and Michael.
Hadley, Aaron, and Michael. Is Michael here? Yeah, Michael's. Looks like Michael's here. Okay. And then I'm assuming Reed, Kyle, and Nathan are all in the same group?
Yeah.
Is that correct? Okay. All right. If somebody else shows up, I'll kick them into the correct room for you. So you guys you're each going to fill out your own copy, but you can decide. Remember, you can share your screen in the breakout room if you want to. You could all do the data together if you want, and then answer the questions together. You can approach it however you wish. I will jump. Remember, there will be a button where you can ask for help, and that will call me to your room, and I will bounce around a little bit and just check on everybody as we go. All right. Any particular questions? 7:44. We'll see where everybody's at around 8:15. If you're not all the way done at 8:15, that's okay. We'll make an adjustment. All right. But we'll see what we can do. All right. And I see Jack is here. We'll get you assigned to your Jack. You're in with who?
I think
You're with Ethan and Mac.
yeah.
Okay. Might take a second, but I'll send you you'll see something pop up that says to join breakout room 4. All right. So I'm going to send you guys out. Any questions before I send you out? Goal is to complete this during the class period. Yay? Okay. So I'm going to open rooms. You'll get a message to join your room. And Jack, I'm going to assign you to number 4 here just a second. Okay, so Jack, you should see a message popping up in a second. Come on, get out of my way. I don't want that. Sorry, somebody's got my screen blocked that I want. There we go. So this is hopefully similar to what you saw in your graph. So up here, we've got the carbon dioxide. And here we sort of have the oxygen levels. Now, because of the scaling of the graph, it doesn't look like there was a significant, which there wasn't. We were having changes in the tenths, but because of the scaling, it looks like pretty much a flat line. Is this similar to what we saw? Yes, no? I mean, your individual data is not going to be the exact same numbers, but you should have seen during the light phase, the CO2 was dropping, and during the light phase, the oxygen levels were rising. And in the dark phase, just the opposite, the CO2 levels were rising and the oxygen levels were dropping. Yes, no? Which this would support. During the light phases, we were decreasing CO2. During the dark phases, we were increasing CO2. Light phases decreasing, dark phases were increasing. Here, it looks like oxygen was fairly steady, but if you look at the fine shifts, it's the opposite. It's the opposite. Not that a bar graph was any better. It's really not the best graph to use, but you see a similar pattern in that process. How do we explain that? What's happening that's causing the CO2 to drop?
Photosynthesis.
You know, photosynthesis does what with carbon dioxide? So let's do this more cooperatively. Sorry, 1 second. Let me stop the share for a second. So if you go to the chat, yes, the chat. Let me rephrase the question. What process is happening that is going to pull out CO2 and increase oxygen? What process is going to reduce CO2 and increase oxygen? And somewhat redundant, but go ahead and fill it in anyway. Which process is going to do the opposite? Which process is going to reduce oxygen and increase carbon dioxide? Because these are complementary reactions. Right? So nothing earth-shattering here, but you can see with the data, we have support for that. Depending on the conditions, that can change. Here we're working somewhat in ideal conditions, but if this plant was in a much warmer environment or in a very humid environment, we might see a little bit of shift in the data, but we'll get to that later. Okay, so where we're going to go next, again, sorry, I'm going to share my screen. Get out of here. Seriously, come on, let me out. There we go. Sorry, everything's not up like it should be because Google wasn't working, so I got to bring it all up. All right. So in a moment, you're going to go to Google Classroom. Let me get to the correct one. Under classwork, there is this Photosynthesis Lab virtual. We just did this one right here, the pivot. And so now we're going to be looking at one of the Darkow simulations. And there's a third lab we'll be taking a look at that's related to photosynthesis once we finish respiration. But anyway, we're going to open up this Darkow lab, and there's this Google Doc that goes with it. All right. So we're going to want to open up the Google Doc. And we are going to want to open up the Darkow lab. All right, so it's called Photosynthesis Simulation. There are a number of controls on the simulation. Those controls are discussed on the first page. Tells you what the different controls do. So make sure you are familiar with the controls so that you know what you can alter. There's quite a few things we can alter. We can alter the presence of light. We can alter the spectrum of light. We can alter the distance from the light. So there's several factors about light we can change. We can alter the pH of the stroma. Remember, the stroma is the dense fluid on the inside of the thylakoid, or around the thylakoids, I'm sorry. The dense fluid is where the Calvin cycle happens. We can add additional NADPH. We can change carbon dioxide levels, temperature levels. And then there's this chemical called atrazine. Atrazine is a chemical that is known to shut down photosystems. It has a negative effect on photosystems. Again, look at the front page and it will tell you what's going on. Just remember, you can go to live feature if you want to try to figure out what something's doing. So what happens if I move further away from the light? Well, it looks like if I get too far away from the light, the reactions shut down. The closer I am to the light, it looks like the more active the reactions are, for example. So that's one easy way to figure out what something's doing. What happens if I go up in temperature? Starts to flatten out, and then as I get higher and higher, it looks like it starts to shut the reaction down. What happens if it gets too cold? Same idea. So if you want to try to figure out a variable, one way to do it is to go on live and you can manipulate it and see what it does. So up here, you're looking at all 4 variables. You can see the code, the legend right here for all 4 variables. Down here, there's tabs. You can look at one variable at a time. So here you can focus on one variable at a time. Glucose, ATP produced, NADPH produced, and oxygen available. So those are the 4 parameters you can measure using this simulation, right? This one requires a little bit more thinking in the process. Understand if you put it on dark, that just simply means there's no light available. If you go to spectrum, you can change the spectrum by wavelength. So here we're in the red spectrum. We can move it into the orange, yellow, green, et cetera, et cetera. So those are all variables you can change. So if we look at the actual document, there's a review of the light reactions and dark reactions. And then there are several different questions that can be tested. So there's an A, B, C, D. I'm forgetting how far it goes. F, G, H. Okay, so it goes up through H. Is that right? Yes, okay. So give me just a second. So for example, let's just look at letter A together, okay? So letter A is about hydrolysis. Hydrolysis is the splitting of water that happens at the very beginning of photosynthesis. That's one of the very first things that has to happen because water needs to supply you with the electrons, because the electrons are how we transfer light energy into chemical energy. We get these electrons excited. So this is one of the major claims being made surrounding A of the light reaction model is, and that being option A, is that light energy is absorbed by chlorophyll and used for the hydrolysis of water and to create a concentration gradient of hydrogen ions. How could you test the idea that solar energy is required for the hydrolysis of water during photosynthesis? So that's the question you want to answer, right? Is solar energy required for the hydrolysis of water during photosynthesis? So I want to go back to my simulation and okay, what can I manipulate here to show that without sunlight, you're not going to hydrolyze water. You have to have sunlight to hydrolyze water. Capiche? So which of these buttons deals with light? Well, up here, I've got several buttons that deal with light. Right? And if I go back to the question, is solar energy is required for the hydrolysis of water during photosynthesis. So which of these products is a direct result of that? Which of these products is a direct result of water being hydrolyzed? And what they want you to recognize is, well, if water is being hydrolyzed and we're producing ATP, we also must be producing NADPH. And so the factor I would probably focus on is NADPH being produced. Because if the only way NADPH can be produced is if water is being hydrolyzed. Am I making sense? So you have to figure out which of these 4 products is going to help you answer the question. I could also do ATP produced. I could do both. Okay. So I'm not making ATP if I don't have water being hydrolyzed to provide the hydrogens to create the hydrogen gradient. And I'm not going to make NADPH because the hydrogen is not being shuttled out of the thylakoid membrane. So those are the 2 factors I would probably look at. So here I can see with the full spectrum, this is what made reset. With the full spectrum, this is what I get in terms of ATP. This is what I get in terms of NADPH. So that means water should be being hydrolyzed. And I can see if water is being hydrolyzed, I'm getting pretty good ATP production. And I'm also getting NADP production. Yes or no? So which variable do I need to change in this case? Well, is light really necessary for this? So let me turn the light off. What happens if I have no light? Do I still get production? And so I can see run number 2, Zippo. I got no ATP if there's no light present. And Zippo, I've got no NADPH. And this would provide the evidence that without light, I must not be hydrolyzing water because I'm not able to produce the ATP and the NADPH. But if I have the light present, bingo, I've got the electrons and hydrogen I need to produce ATP and to produce NADPH. Am I going anywhere there or am I losing you? Blank stares don't help me. Any clarifying questions, you can throw it in the chat or you can. So then I would I could recreate those graphs. And I would put a line on here. If it was me, I would make a graph and I would show the production of ATP and I would show the production of NADPH with light and without light. So I'd have 4 lines on here. The up and down line for ATP and the flat line for ATP, the up and down line for NADPH and the flat line for NADPH. That would be showing that light is necessary for the hydrolyzing water. So then 3 is this idea that solar energy is required for hydrolysis of water during photosynthesis supported by the simulation. Yes, because, and then I would give some of the data. Because when light was present, we produced anywhere from blank to blank amounts of ATP, and we produced anywhere from blank amounts to blank amounts of NADP. When no light was present, we produced 0. So why is the hydrolysis of water required for photosynthesis? It supplies the electrons, which pick up the sun's energy to create the proton gradient, which allows us to create ATP and NADPH. I might give a little bit more detail than that, but that would be the idea. Yes, no. Trying to give me an idea how you would work through it. Or I just don't even know what to ask. So really the key is the key to this whole thing is recognizing what's being produced, and by measuring that, how do I answer the question? Which of these 4 products is going to help me answer my question? And then what variables do I have to manipulate to see how that works? So in this case, I had to understand when you hydrolyze water, that's an input. The output's going to be NAD, pH, and ATP. And then the variable that we were testing was light. So I had to understand what my inputs were, I had to understand what my outputs were, and which variable could affect that. And that's how it's going to be for all of the questions. What's my inputs, what are my outputs, and which variable am I going to alter to show whether that works or doesn't work? Anything? All right, so I'm going to assign each group 2 of these. Okay, each group 2 of these. Yes. You each will complete the paperwork for yours, but you can certainly work together on how you want to attack it. Okay, so group 1 will be doing A and E. Group 2 will be doing B and F. Group 3 will be doing C and G. Group 4 will be doing D and H. Group 5 will be doing B and G. Group 6 will be doing C and F. And group 7 will be doing D and H. Everybody got that? Okay. Holy cow. Is it really that late? Okay. So we are at 8:40. I'm going to have you go to your breakout rooms, just so at minimum you can touch base. You can divide and conquer and get back together. You can decide on a time that you're going to meet between now and Thursday. You can decide you're each going to work on it yourself and then check back with each other later. You can each decide we're just going to complete it by ourselves and then we'll check each other at the end, however you guys want to attack it. But you might want to at least look at the 2 problems and think it through while you got all of you together. And again, feel free to schedule your own meeting on Zoom or Google Meets or Google Hangouts or FaceTime or any other of about 60 different apps you can use. Feel free to schedule your own time to meet as well. But when you come back on Thursday, you'll need to have submitted the lab you were just working on and this one. Yes, no, excuse me. Wow, excuse me. Corona. All right. There will be a brief check quiz, and I mean brief, like 10, 15 max, 15 absolute max questions just on the basics of enzymes and photosynthesis, just to see if you're getting the basic ideas about them. Look at the learning objectives at the front of the digital notebook. Yes, that should help you. And it's just a quiz, and then we'll continue to move forward. I haven't decided exactly what we're going to do to move forward. In terms of work, whether we just take the break and do nothing, or we take the break and try to do a small amount. I'm still convinced if we do a small amount and break, it's going to be easier on you in the long run. But it just depends on where you're at mentally. So I'll probably send you a survey on where people are at in their thought process. All right, so anyway, I'm just rambling and thinking out loud. I'm going to send you to the breakout rooms. Take some time to look it over, look at the two that you got, talk about it, discuss it. I will bring everybody back to the main room in about 10 minutes for a final dismissal and just a final check-in. Yes? Okay, you'll see an invite to your breakout room coming back up. Oh, Aaron is now here. Okay. Aaron, I'll be sending you a link to your room 6. So you should see something to join room 6 coming up, Aaron.